Bacterial estimation based on cell mass has both direct and indirect methods. Under the direct method, we have the dry weight determination, and under the indirect methods, we have the turbidometry and the determination of nitrogen or protein content estimation. So, methods totally these methods are used to quantitate the cell mass. So, here the cell mass, the weight of the substance or the material to be estimated will be taken, and this estimation will be either directly taken as in the case of dry weight determination, or that will be indirectly taken. So, in turbidometry, we usually get the turbidity of the solution, and this turbidity is an indirect measure of the cell mass, and also the nitrogen or the protein content present in the system, present in a particular culture, will give you an indirect measure of the cell mass. So, first see the dry weight determination, and this is, as the term indicates, this is the most direct approach for quantitative measurement of the mass. This is the dry weight or the dry mass of the cell. Usually this is applied to to multicellular organisms than bacterial cells, because bacterial cells are very microscopic, and taking or the measuring the weight is a laborious task. But still we will be listing this one as a method. So, in this method, the sample is centrifuged, usually centrifuged or filtered, and dried in an oven at around 60 degrees Celsius for a short period of time. Then this material is weighed, or the weight of this material will be taken in a weighing machine, right? And definitely the standard protocols will be preferred, like the weight of the filter paper will be taken out. That means that will be minus from the total weight. Okay, so this is what how we do the dry weight determination. And from this one, if you want, you can convert this mass into number, if you know, or if you can find out what is the weight of a single cell, then you can from that point you can find out what number of cells will be there in that particular sample. Yes, and in the indirect method, the turbidometry is a very common method and very usual procedure in laboratories. It is the cheapest procedure to estimate the bacterial growth. This can be detected by means of instruments like a turbidometer, nephelometer, colorimeter, or spectrophotometer. These are the various examples for detecting the turbidity of a solution. Right. So, we know that after inoculation into a test tube or into a broth, normally the cells will multiply and the number will increase, and as and when the number increases, the turbidity of the solution also increases. So, it is having a direct or the proportionate, the turbidity of the solution is proportionate to the concentration of the cells present in that particular solution. Usually this idea works under the Lamberts law. That means the turbidometry works under this concept. If the number of cells are more, the turbidity is higher, and if you have more turbid solution, the light passing through that will be less due to scattering of light rays. This is the idea of Lamberts law. So, the ability of the culture to block the light can be expressed as either percentage of light transmitted through the tube or the amount of light absorbed in the tube. So, these are the two possible determination. The percentage of light transmitted is inversely proportional to the bacterial concentration. Similarly, the absorbance is usually we denoted as the optical density is directly proportional to the cell concentration. So, that value will be obtained. So, higher the turbidity of a solution, you will get a particular OD value that also will be higher. So, lower the turbidity, the lower will be the OD value. Okay. In case of if it is a percentage of transmission, it will be inversely proportional. That means if you have a higher turbid or a more turbid solution, then you will have a lesser value for percentage of transmission. So that is how you use this machine. Now we have another machine called a Nephelometer, or the technique is called Nephelometry. This also having almost the similar concept, but the difference is that the measurement of this in this machine, the measurement occurs for the scattered light instead of the absorbed or transmitted light. The scattered light will be measured. Okay, so the idea is that a dilute solution of small particles will scatter light passed through it rather than absorbing it. So with that concept, this machine has been designed. So the amount of light scattering from the sample will be directly proportional to the increased concentration of cell suspension. So as the scattering increases, which says that, indirectly says that the cell concentration is higher, right? So once in a condition, if a substance is completely dissolved in a solution, then what will be the value? There will be no scattering of light, and hence there will be a very low nephelometric value, right? So the scattered radiation is measured. Usually the detector is located at an angle of 90 degrees to the sample. You can see this image so that you will get an idea. So the scattered light from the sample will be detected, and the detector is located 90 degrees to the sample. And usually this machine or this apparatus is widely used in immunological reactions or serological tests rather than in microbiological enumeration procedures. The determination of nitrogen content or the protein content is an indirect way of measuring the cell mass, because proteins are the major constituent of every cell, and so is nitrogen also, because nitrogen is part of a protein. So it is possible to measure a bacterial population or the cell concentration in terms of bacterial nitrogen or bacterial protein. So if you take nitrogen, the nitrogen percentage per cell is usually around 14. That means there will be 14 percentage of nitrogen on a dry weight basis per cell, right? So what you have to do is harvest the cells from a culture, wash them free of medium, and then perform a chemical analysis for nitrogen. Then you will get a particular value. That means this much percentage of nitrogen is present in that particular sample, and from there you can predict that this much number of or this much quantity of cells are present in the sample. So this is the way. So one of the there are various limitations, like the nitrogen determination is laborious and can be performed only on specimens free of all other sources of nitrogen, right? There should not be any other source than the bacteria. Furthermore, the method is applicable only for concentrated populations, and usually these methods are, because of these limitations, not widely used for normal or routine laboratory procedures, but instead it's used for research purpose. Now we are moving into the final concept, what is called the estimation of bacteria based on cellular activity. And here in this one we have only the indirect methods. Here the basic idea is that a particular cell perform a particular activity, and if 100 cells perform that activity, the magnitude of that activity will be, and if the magnitude of the activity is detectable, then we measure the magnitude of that activity and from that value we'll find out the total number of cells. Okay, that is the idea. So for that we can go for different products or different substrate like acid production, alcohol production, carbon dioxide release, and even the consumption of certain substrates. All those are different activities. So it is like the specific changes caused in growth medium as a result of the activity and multiplication of bacterial cells, and the majority of the reactions are enzymatic. So the enzymatic reactions will be detected. We can find a lot of examples like detecting cell products such as acid and gas by the bacteria, and that could be counted and that will be converted into the cell number or cell mass. Then we have a representative example is methylene blue reduction test or methylene blue reductase test. So here usually this MBRT test is used for to assess the quality of milk, right. MBRT test is a test for assessment of quality of milk. Here actually the bacterial load is detected, right. Consumption of oxygen in milk is detected through the reduction of the dye methylene blue. Removal of the oxygen from milk and the formation of reducing substances during bacterial metabolism cause the color to disappear. That means methylene blue will be colorless when the media doesn't contain oxygen, right. So the removal of oxygen is proportional to the growth of bacteria in the milk. So when bacteria is more, the oxygen removal will be faster. So greater the number of bacteria, the quicker will be the oxygen consumption, and in turn there will be fast disappearance of the color of methylene blue, right. So the time of reduction is taken as a measure of the number of organisms in milk, and that will be used for estimation, right. So this is the way how we use this activity. Here we use the oxygen consumption activity, right. The bacteria consume oxygen, and that activity will be detected here with the help of a oxygen reduction oxidation reduction dye called methylene blue. Okay. So by that we wind up this particular session of bacterial quantitation. So here I am summarizing this as direct methods. We have Petrov-Hose chamber counting chamber, then Coulter counters, filtration and staining, dry weight determination are the direct methods. And under indirect methods we have discussed plating methods like pour plate and spread plate, then membrane filtration followed by plating, then MPN method, turbidometry, under which we have turbidometry like colorimetry and spectrophotometry. Then also we have nephelometry, then nitrogen content or protein content determination, then cellular activity. So these are the direct and indirect methods. Okay. So these are the various references you can refer for further discussions, and thank you for listening this presentation.